Hello there, my marvelous microbiology students. We have done a lot in this class in terms of identifying organisms. We've done the stains, we've done the biochemical tests, the differential media, and all that kind of stuff. One other thing that is used to identify microorganisms is to look at their oxygen requirements. Now, it's been a while since we've talked about oxygen requirements, but remember that bacteria or any other microbe have different oxygen requirements depending on what environment they live in. It's hard for us to imagine living without oxygen, but oxygen can actually be toxic. So there are toxic forms of oxygen. And it's these toxic forms of oxygen that are detrimental to cells. Most cells have certain enzymes that break down these toxic forms of oxygen into compounds that are perfectly harmless, such as water and regular oxygen, O2. So we're going to look at bacterial requirements for oxygen. Now there's 5 major categories for oxygen requirement. The first one is your aerobes, and you can call them obligate aerobes, meaning that they need oxygen, okay? The other group are going to be the facultative anaerobes. These guys are the ones that like oxygen, but if oxygen is not present or the amount of it is limited, they will start to grow anaerobically. Another group are the microaerophiles. These require very, very low amounts of oxygen, and if the oxygen in the atmosphere is high enough, it will actually slow down their metabolism. It won't kill them, but it will slow them down. As opposed to the obligate anaerobes, those are the ones that literally do not have the enzymes to deal with these forms of oxygen, and they will die in the presence of oxygen. The last one are the aerotolerant anaerobes, and these are the ones that, like it sounds, they can tolerate tiny, tiny amounts of oxygen. So those are the 5 groups that we're looking at. And if you refer to your lab manual, you'll notice that when you grow them in tubes of either a solid medium or in a reducing medium, they're going to grow at different levels. If they're aerobic, they're going to be growing at the top. If they're anaerobic, they're going to be growing at the bottom. If they're like a facultative anaerobe, they're going to grow at the top and then a little bit less as it goes down. And you can see the growth patterns of the other 2 as we look at that. So what you're going to be doing this first oxygen lab is you're going to be taking the organisms, and I forget, I think there's 6 of them, and you're going to have 6 tubes. So you'll have 6 tubes that are, oh darn it, these are both solid. Pretend one of these is not a solid. This is a liquid, okay? All right, so you're going to have a media called brain heart infusion media. So it's really, really enriched, okay, so they love it. And what it is, it's an actual medium of agar. But when you receive it, it will be in a beaker of hot water. It's been boiled, it's been autoclaved, and it'll be taken from the autoclave and then put in this warm water. Now there's no water in here. And that warm water will keep it at about 45 degrees. So you don't want it so hot that when you put the bacteria in there, they're boiling. And basically, if it's warm to the touch and you can still touch it, then it's cooled off enough, okay? So when you pull them out of the water, it shouldn't be so hot that you can't touch it. And then we'll also put a thermometer in the water to make sure that it's around 45 degrees. Because if it gets too much cooler, it's going to start to solidify. But too warm, it's going to denature the enzymes of the bacteria. So what you're going to do this time, and then this right here is a beaker of ice. So this time you're not going to use the inoculating loop, you're going to use a sterile pipette. And so you'll using the sterile pipette like we've done before, you're going to draw up just a little bit of sample, it doesn't need to be a lot, of that particular test subject. And so you're going to draw up a little bit, and this is a liquid, remember? Okay. And you're going to drop a couple of drops of the culture into the liquid. Now before you draw it up, remember that bacteria have a tendency to grow on different levels of the media. So before you draw up your sample, go ahead and draw up, push out, draw up, push out, and kind of mix it a little bit before you grit your sample. So you're not maybe getting just a little tiny bit, and you want to mix it a little bit. Remember to do that gently so it's not splashing out. And then you're going to take your couple of drops and put it in the liquid that you've taken from the water bath. And after you do that, you're going to take it between your hands. Now once again, I would be wearing gloves, I would be wearing my goggle, all sterile technique would be done, but we've been through that 100 times, so we don't need to go over it anymore. Make sure that the lid is on, and you're going to run it between your hands, and your intent is to mix it with the liquid, okay? So after you've done that, you're going to put it directly into the ice for about 10 minutes. Say place into, it doesn't have a time, so you put it in just till it starts to solidify, okay? And so you want it solidified because the idea of this is you want to be putting that solid medium into the incubator, and because it's gelatin, remember, it'll stay a solid. So once it's solidified, you should have the bacteria stuck within the media, as opposed to when we did the stab and we pushed it down into the media and it maybe grew out. This should be all in the media. So when they start to grow in the incubator, depending on their oxygen requirements, they're going to grow toward the top, toward the middle, toward the bottom. So after you've done that with all 6 of them, so you'll have a beaker that will hold them pretty good, and your tubes will probably be a different size, not this size here. Then you're going to take them and put them in your rack after they've solidified. So once again, I thought it was 10 minutes, but there's no time period. So once they've solidified, you put them in the incubator.